So welcome everyone to our Color Research Society of Canada Kaleidoscope lecture series. Very excited to welcome Ryan today. I'm just going to begin with our land acknowledgement. The Color Research Society of Canada's activities related to sharing color knowledge take place across Canada, understood as part of Turtle Island, the ancestral homelands of over 630 First Nation communities, representing more than 50 Indigenous nations and languages. We work to respect, affirm, and support where necessary the inherent treaty rights of all Indigenous peoples across these lands and waters. Our commitment to honoring the knowledge, ways of knowing, and wisdom of Turtle Island's Indigenous peoples aims to contribute to the decolonizing work required today for moving towards a just society. So welcome everyone. My name's Robin Kingsborough of the CRSC, and we are the Canadian arm of the International Color Association, the AIC. The CRSC is a nonprofit organization for color research focused on fostering a cross-disciplinary sharing of color knowledge. We seek to develop and support a national cross-disciplinary network of artists and designers, scholars and practitioners with an interest in engagements with color, and to encourage discourse between art, science, and industry related to color research and knowledge. Can I ask everybody to put their mics on mute? And I'm very happy to welcome our speaker this evening, Ryan Schott, who is from York University. A few of us are from York University here, and very much looking forward to finding out about his work. And I just hit the wrong screen, just one sec.
All right. Well, thank you, Robin, for that.
Let me just give you your bio. Sorry, my screen just disappeared once. So Ryan is going to talk this evening about the molecular basis of color vision and its variation about vertebrate animals. Ryan is an assistant professor at the Department of Biology and Center for Vision Research at York University in Toronto. Ryan completed an MSc at the University of Toronto, studying the systematics and ontogeny of dome-headed dinosaurs. That was always my son's favorite. And a PhD, also at U of T, studying vertebrate visual systems evolution using transcriptometric, genomic, and computational approaches, with a focus on the evolution of photoreceptor transmutation in snakes and geckos. After his PhD, Dr. Schott joined the Smithsonian Institution's National Museum of Natural History as a postdoc, where he studied the molecular evolution of visual systems in frogs as part of a joint NSF and ERC funded international collaboration. At York, Ryan has a lab where he seeks to understand how organisms adapt to distinct environmental conditions, how novel cell types can evolve, and the role of developmental changes in evolution using the vertebrate visual systems as a model. I'm very happy to welcome Ryan this evening. So please, we are very much looking forward to your talk on the molecular basis of color vision and its variation among vertebrate animals. Take it away, Ryan.
All right, thank you very much for that kind intro, Robin. Let me just make sure I click the right share sound feature here again. I think I got it. All right. Well, I was going to do a bit of an intro. You covered a lot of that for me, which is fine, which is great. But I was going to start off with a bit of a little bit of a trajectory of my research career. So, I was born and raised in Toronto, so I'm kind of from the area where I'm now lucky enough to get to work. And I did started my kind of academic career as an undergraduate at the University of Toronto and the Royal Ontario Museum. And here's a much younger version of me when I was doing some field work in southern Alberta. And as Robin mentioned, studying these dome-headed dinosaurs. And so I was interested in how these animals had evolved and how they're related to each other and kind of how they grow, which is not a trivial thing to figure out when all you have to study is a small bit of fossil bone, in this case, the top of the skull. But I became more and more interested in genetics and molecular mechanisms, and so this was not a great system to get at that. So I moved on to do a PhD in the Chang lab, And this is a vision lab, but I was interested in the genetics of these horned lizard horns because this is a group of animals I had been studying as part of an undergraduate side project as like maybe a cool analog to some of the ornamentation in dinosaurs like Andes dome-headed dinosaurs or in things like Triceratops. But I was in a vision lab, and so I quickly became fascinated with the diversity of animal vision, and in particular, in these cool systems in geckos as well as in snakes. And so that was a lot of the focus of my PhD. While I worked on some other vertebrate animals like mammals, such as bats and fishes like these cichlids here. And then from there, I moved on to the Smithsonian National Museum of Natural History, where I joined the Bell Lab and started to work on amphibians and in particular frogs and their visual systems. From there, I moved to York, and I've been there for a few years now, working in my lab, setting things up, getting everything going, and having a few students start off a few different projects. And so we're really focused on visual system diversity and how that influences how these animals can evolve and adapt to their environment. So here's my lab at our in one of our annual lab barbecues, including a couple of the junior lab members there. And kind of right off the bat, a few of the um, images and things I'm going to show are part of the work I did as a postdoc and kind of continuing on since then with what we call the Frog Vision team. And so we put together this ebook about frog vision and vision in general. And so I just want to kind of acknowledge the co-authors on that right off the bat, as well as, of course, all of our funding and institutions. All right, so that was a bit about my academic trajectory. So I want to give you a brief overview of my research interests, which are really driven by natural diversity. And this diversity is largely a result of the different environments that organisms inhabit. And so these can kind of see from the images here, these environments are quite different from each other, and these favor different characteristics for reproduction and survival. Right, so I'm broadly interested in how organisms have adapted to their environments, and in particular, how changes at the molecular level, which could be things like changes to protein structure and function, or gene expression, or developmental timing, so how development changes across species, how these things can kind of contribute and produce phenotypic diversity and evolutionary novelty from the cellular level through to the organismal level. And so we use a number of different techniques in the lab to study this, including comparative genomics and transcriptomics, different molecular evolutionary methods, as well as imaging and functional assays. And where lab is focused on the visual system, but why the visual system versus all the other different things we could study. And so one of the great things I think about the visual system is this direct link that it provides between an organism and its environment. And so this could be things like finding food, finding mates, or avoiding predation, among many other things. And so just returning to this little collage of different environments, I think you can see the way they drastically differ in their spectral properties. So you have these kind of red tinged waters in the top right there versus blue water. You have under the forest canopy where a lot of the green light is being reflected. And so there's a lot of variation in the light availability, and that's led to this wide diversity of visual systems that we see in animals, and in particular, I'll focus on vertebrate animals today. And so kind of broadly, I'm interested and my lab is interested in how did this diversity evolve? And how do different molecular and developmental mechanisms contribute to this diversity? But what I'll focus on for today is how do animals actually see color, and how does color vision vary and evolve in different vertebrate animals? But before we can talk about color and color vision, we do need to just review some of the basic properties of light, right? So without light, we can't see. All right, so light has a few different key properties that we'll kind of touch on here. So intensity, so how bright is the light? The wavelength of light. All right, so what's the wavelength, which is going to be related to their color, as we'll see, and you can already kind of take away from the rainbow image that I'm sure you've seen many times before, as well as the direction of light and its polarization. So if you've ever worn polarized sunglasses, you'll know that those lenses are cutting off any light that's not oriented in a particular way, thus reducing the amount of light that can reach your eye. But since we're going to focus on color today, we will be focusing on that second property of wavelength. And we can see, just a kind of blow up of that kind of rainbow of colors going from the ultraviolet on the right there to the infrared. Sorry, the ultraviolet on the left there to the infrared on the right, kind of sandwich in there is what we call the visible light spectrum. Although what we find visible is not necessarily the same as what animals will find to be visible. All right, so a bit about that more in a moment. We also need to think about how light moves through and interacts with the environment. And so we have a little image here showing light coming down from the sun. And we can notice a few different things here, ways that light can interact with or propagate in the environment. So it can refract, meaning it's changing direction. It can scatter, meaning it's pushed off in all different directions. It can be absorbed. So here the light is being absorbed by the frog and the kind of red, orange, blue, and violet range of the spectrum. And then some light might be reflected. And so here, this is obviously an oversimplification here, but reflecting the green light of that green frog to the eye there of the snake. So we might want to ask ourselves, what is color? I'm talking about, Color Research Society. I'm sure it's a kind of hot topic. And, it's not necessarily something we can simply answer, with a simple definition. And there's going to be a lot of debate and, philosophical considerations here. But we'll just kind of break it down into two different things, at least for today's talk. So is color a physical property of an object, or is it a feature of our perception of objects? And we'll kind of see today it's definitely a little bit of both. So we can think of color as a physical property, being what kind of light is absorbed and reflected by an object. So here on the graph, I'm showing the reflectance, so what light is reflected off these different types of leaves. And so you can kind of see in the green curve here, the darkest leaf, it is reflecting in the green, although it's low because it's a dark leaf. Whereas, our kind of brighter green leaf is reflecting more, same with the yellow. And then the red is kind of showing like this. So it's not necessarily just reflecting red because it's a red leaf or just reflecting green because it's a green leaf. It's a lot more complex than simply, the rainbow spectrum on the wavelength of light would suggest. Now, part of what's causing the differences in reflectance between the leaves is the chlorophyll and the carotenoids in the leaf, which are absorbing light, although it's not all of what's causing those colors. There's other things in the leaves that are reflecting light, like the cellulose. But we can see that the chlorophyll and the carotenoids are not absorbing light in the green range, but they are absorbing the blue and violet and orange and red ranges of the light spectrum. And so this is some leaves. We can also look at a different animal and how its body pigmentation reflects light. And so this is an example of a frog, Biturana uranoscopa, and it reflecting in the dorsal and ventral part, so the top and bottom of the body. So here's the ventral reflectance, so it's a lighter color of green, and so it has a higher reflectance. And this is the dorsal part, the top of the body. And so you can see it's also reflecting a lot in the green and yellow range of the spectrum, but at a lower level because it is a darker color. This is actually some work from some data from our ongoing study on frog vision, and my undergrad student Hasti has been one analyzing some of this reflectance data and trying to look at how these reflectance patterns change across species and maybe how that interacts with how frogs can see each other and how predators can see the frogs. And this is still kind of in an early stage, so I'm not going to show too much, not really show any of this more today, but it is something that we're kind of actively working on. Now we could also think about color as perception, right? So you may remember this from probably 10 years old now, this kind of viral image that went around. Is this dress blue and black, or is it white and gold? And depending on your personal color perception sense, depending on your screen that you're looking at this on, you might see it as one versus the other. And it has to do with the background light conditions. And so this is a little GIF showing switching between the two. For me, it still looks blue and black no matter which way I look at it, but definitely remember talking about this as a grad student with other people in the lab and, having arguments about this, and people did it all over the internet back then. So definitely is also a part of how you're perceiving color. All right, so with that kind of out of the way, we can think about how animals are going to be able to detect color. So of course, they need to use their eyes to detect the light. And so we can think about different types of eyes. So eyes have evolved multiple times in animals. And there's probably at least 10 different types of eyes, ranging from these simple eye spots here in the top left up to these kind of more complex eyes. This is a camera style eye, and it has a lens. Now, the lens is an important aspect here because it allows us and other animals that have camera type eyes to focus light onto a focal point, being where the light sensitive cells in the eye actually are. So when you have a distant light source, the light rays are going to be parallel to each other, but you have a near light source, the rays will be diverging. And so that'll push the focal point behind where you might want from a distant light source. Now, the way we can get around that is by changing the shape of the lens or by moving its position within the eye. And so this allows us to maintain a crisp image. And is one of the ways that vertebrates that have camera style eyes and other animals with these type of eyes have been able to get high acuity vision, meaning high resolution vision. All right, so here's a sketch of a human eye, and then a more simplified version of a frog eye. Now you can see that they're relatively similar, so a lot of the main structures are conserved across vertebrates. And the kind of key ones we'll take a look at today, which we just kind of saw the purpose of the lens there. We'll look at next the retina. So the retina is actually at the back of the eye, and it's the light sensitive part of the eye, but it is this multi-layered neural tissue. So it has multiple cell layers, including the light sensitive cells, which are the photoreceptors, which are actually at the back of the retina. And so you can notice that the light is coming into the eye this way, but that the signal is actually being processed that way. And so this is maybe backwards to what you might kind of expect if you were to design yourself an eye. Now, what does the retina actually look like in not, cartoon form? So this is a light microscope image of a slice through the retina. You can see the photoreceptor cells here. And then on the bottom here is a scanning electron microscope image. So it's a bit higher resolution. It's not necessarily sliced through, but here we can see the kind of photoreceptor layer on top. Okay, so just returning to this idea that light's going this way, but the signal's being processed in the opposite direction. Right, so we have the photoreceptors here, the light sensitive part of the eye, kind of at the back of the retina. So again, this is not the way you might want to design an eye, and actually is not the way that other types of animal camera type eyes are set up. So here is the eye of which you might find in a cephalopod, so an octopus, squid, or the nautilus shown here. And they actually have the photoreceptors at the front. And so what happens in a vertebrate eye is you end up with this blind spot here, where you have the optic nerve leaving the eye, sending that signal of light to the brain. And in cephalopods, you don't have that blind spot because it's flipped around the way that maybe makes a bit more sense intuitively. All right, so let's go back to the retina, and we're going to talk a bit more about these photoreceptors. All right, so we have two types in vertebrates. We have rods and cones. And again, these are the light sensitive cells in the eye. All right, so here's another diagram of them here and what they actually kind of look like. These are mammalian cells, I believe. Again, from just a light microscope image of what a cone looks like here on the left and what a rod looks like on the right. So pretty close to what we actually are kind of depicting and drawing them as here. All right, but why do we need two types of photoreceptors? All right, so rods, at least in the human eye, are about 20 times more common than cones, and they're highly light sensitive. Right, they're highly light sensitive, but they are prone to becoming saturated, meaning that if there's too much light intensity, they'll stop functioning. And so because of that, they really only work under dim light conditions. They also react a lot more slowly than cones do, and they take longer to recover. All right, so they're really good for dim light vision, but they're not going to function under daylight. Now, the rods absorb visible wavelengths with a peak around 500 nanometers, and there's only one type of rod in most vertebrates, including ourselves. And so rod vision is in shades of gray. Now, cones are less sensitive to light than rods, but mostly this is a good thing if we're using them during the day when there's plenty of light available, so that way they can have faster reaction times as a kind of trade-off, as well as provide highly detailed vision. So this high acuity vision in these small receptive fields. Now, most vertebrates, at least, have multiple types of cones from 2 up to 4 or more. We'll take a look at some of that variation in a bit. But these different types of cones actually respond to different, or at least respond maximally to different wavelengths of light. And so this is what allows us to produce color vision. All right, so if we take a look at the photoreceptor and we kind of blow up the top part here, we call this the outer segment. And so within this outer segment are these discs. They're made by cell membrane, plasma membrane. But in these discs is actually the light sensitive molecules in the eye, which we call visual pigments. And so there's a representation of that there. And so a visual pigment is a combination of an opsin protein as well as a light sensitive chromophore. Now you can't really see the chromophore in this image here, but the chromophore is called retinal and it's based on vitamin A. And it's the chromophore that's actually light sensitive and that actually changes its conformation when it absorbs light. So I have a little animation here of what that looks like. So here's our visual pigment and the chromophore there is in orange in the middle. And in a second here, light's going to be absorbed by the pigment, causing that chromophore to photoisomerize, which then causes a conformational change in the opsin protein. The chromophore then also exits. And so in order to detect a new light signal, we actually need to get a new, what's here, the original conformation, which is called 11 cis retinal. We need a new one of these molecules to come in to reset that. But it's this conformational change in the protein that actually triggers the biochemical pathway downstream of the visual pigment that eventually results in an electric signal being sent through the retina and then to the brain. Okay. All right, so how does that process work? We call this phototransduction, and I have a couple little videos here that I worked on with some colleagues to kind of explain this process, using some frogs as a little example here.
As with all animals that have vision, frogs use eyes to detect light signals in order to develop an image in their brain that then allows them to interact with their environment. In this video, we will take a closer look at how eyes detect light and transmit an electrical signal to the brain. This process is called the phototransduction cascade. Remember that vertebrate eyes contain a retina, and this is where the light-detecting cells called photoreceptors reside. There are two types of photoreceptors, rods and cones, and for this video we will use a rod cell as our example. The outer segments of photoreceptors contain membranous stacks that house the visual pigment and other players in the phototransduction cascade. Light enters the eye and will excite the photoreceptor, and this results in the initiation of the phototransduction cascade. The primary consequence of the phototransduction cascade is a signal being sent to the brain. So let's take a closer look at this phototransduction cascade. In addition to the visual pigment, there are several other proteins and protein complexes that play an important role in the phototransduction cascade. Some of these reside in the disc, but also in the cell membrane, which provides an avenue for the inside of the cell to communicate with the outside of the cell. Now don't worry, we won't discuss all of these proteins now. We will introduce them when we come to their role during the phototransduction cascade. In addition to proteins, we will see that the cell responds to fluctuations in the concentration of small molecules and ions, most importantly calcium ions and a molecule called cyclic guanosine monophosphate, which we will call cGMP for short. Changes in calcium concentration causes polarization across the cell membrane, so we will keep track of how these change along with cGMP during the three major stages of the phototransduction cascade, which include the dark state, during which the visual pigment is not stimulated by light, the light state, during which the visual pigment is stimulated by light, and the recovery phase. So let's start with the dark state. The dark state. In the dark, most of the phototransduction proteins are inactive, except for the cyclic nucleotide gated channel, or the CNG, and the light insensitive sodium-calcium-potassium exchanger. The exchanger is found on the outer membrane of the photoreceptor and functions to continuously move calcium and potassium out of the cell in exchange for sodium into the cell. The CNG is likewise found on the outer membrane and in the dark state is bound to cGMP, which causes it to move sodium and calcium into the cell. The combined action of these two proteins results in high intracellular calcium levels and an overall influx of positively charged ions, causing a depolarization current of about -30 to -40 millivolts, known as the dark current. Let's take a look now at what is happening at the synapse of the photoreceptor during the dark state. The depolarizing dark current activates calcium channels at the photoreceptor synapse. These channels bring calcium into the synapse, which results in continuous release of the neurotransmitter glutamate in the dark. Glutamate maintains the dark state, and the brain does not receive signals from the retina. In summary, in the dark state, high calcium concentration causes the continuous release of glutamate that prevents the brain from receiving signals from the retina. The light state. Activation of a visual pigment by light induces a conformational change in the visual pigment, allowing it to bind to the protein transducin. Transducin is composed of 3 subunits, and binding to the visual pigment causes the release of the active subunit from the other 2, allowing it to bind to phosphodiesterase, in turn activating it. Activated phosphodiesterase converts cGMP to GMP, reducing cGMP levels, which in turn results in the closure of the CNG. The continued action of the sodium-calcium-potassium exchanger results in hyperpolarization and an increased negative charge of the photoreceptor cell and also a reduction in the calcium levels. Now let's take a look at what is happening at the synapse of the photoreceptor during the light state. Hyperpolarization causes reduced activity of the voltage-gated calcium channels at the synaptic terminal, resulting in slowed or halted glutamate release into the synapse. This results in a signal being sent through the retina, where it is further processed and eventually sent to the brain, resulting in vision. Recovery. After light activation, the photoreceptor cell needs to be reset to its depolarized dark state through a series of deactivation steps, many of which are regulated by negative feedback from decreased calcium levels. In the dark, when calcium levels are high, the calcium binding protein recoverin is normally bound to G-protein coupled receptor kinase, or GRK, in activating it. However, the light-induced decrease in calcium results in the dissociation of GRK from recoverin. GRK preferentially phosphorylates activated visual pigment, which partially reduces the visual pigment's ability to activate transducin. Another protein called arrestin preferentially binds phosphorylated active visual pigment, which completely deactivates the visual pigment. The active subunit of transducin is deactivated by the regulator of G-protein signaling 9, or RGS9, protein complex, following which it can reassociate with the other 2 transducin subunits. The lowered calcium concentration also results in activation of guanylate cyclase activating proteins. These in turn activate guanylate cyclase proteins, which synthesize new cGMP. Increasing cGMP concentration reopens the CNGs, eventually restoring calcium concentration and depolarizing current to the dark state levels. The increased calcium concentration inactivates G-protein coupled receptor kinase and has the additional effect of deactivating guanylate cyclase activating proteins and guanylate cyclases.
Okay, so I hope that gave you a little brief overview of kind of the molecular process of phototransduction, how light is actually transduced from a light signal into a electrical one that eventually gets sent to the brain. Now, there's a lot more that goes on there with processing the signal in the retina, as well as how it's actually perceived in the different visual centers in the brain. We're not going to get into too much of that detail today, but what kind of wanted to touch on is, how can we actually see these different wavelengths of light? So how is that kind of achieved at the molecular level? So we took a look at the absorbance profile of a visual pigment. So we're showing here the normalized absorbance. So what wavelengths of light and how much is being absorbed. This is a kind of pigment that is absorbing most maximally around 500 nanometers in the green region of the spectrum. So this might be the pigment that's found in the typical rod cell of a vertebrate. Now it has this vitamin A1 based retinal chromophore, and it's going to have a particular protein sequence. So the amino acids that make up the protein have particular identities. But if we were to change the identities of those amino acids, if we'd actually change the components that make up the protein, we can shift the absorption spectrum. And so if we get this to go here, if we change a couple of these amino acids here, make two different substitutions, two mutations, we could shift that absorbance profile over to the right towards the red. And similarly, if we change some other substitutions, it could even potentially just be a single site, which is the case of this shift from violet to UV sensitivity that's found in birds. We can get this kind of shift in sensitivity going the other way towards, we call it blue shifting, although it's to the violet in this case, or the UV. We also can use different types of retinal chromophores. So this one is based on vitamin A1, but there's another one that vertebrates use that's based on vitamin A2. And so if we use that one, and it has this extra double bond here, so it's a different chemical structure. If we use that one, it also shifts the absorption spectrum, and in this case, it goes towards the red as well. If we go from the A1 based visual chromophore to the A2 based. And so ancestrally in vertebrates, so we think that kind of the ancestor of all vertebrates would have had 5 different types of visual pigment encoded by 5 different opsin genes. And so it would have a kind of the one main rod one here, as well as 4 different cone ones that are each sensitive to different portions of the light spectrum. And so you can see these don't kind of match the order of colors here. These are the order of their evolutionary relationships, but they would have had each of these 4 types and possibly been able to distinguish between these colors in a tetrachromatic color space. And so if we just focus on the cones for now, it would have looked maybe something like this in that ancestral vertebrate. But what about us, right? So we only have 3 types of cones. We have blue cones, green cones, and red cones. And so how do we see or how do we perceive color? And so if we think of taking just some pure kind of blue light around this wavelength, it's only going to activate the blue cones, right? So 100% of the cones activated would be blue. And so we would perceive this kind of light blue color. In the middle here, we have a light that's kind of a yellow color in this wavelength range. It's activating both our red and green cones. Sorry, we're doing green here. It's activating our red and green and our blue cones, but the green the most, and that gives us the green color. The yellow one is activating both red and green equally. That's our last one here, the 83. The percentages there, aren't too important to worry about. But these kind of combinations is how of how much of one pigment is activated versus another is part of how we perceive differences in color. And so we would call this system where we have 3 types of cones a trichromat, and our vision would look something like this. So this is us as well as some other old world primates, whereas many mammals are actually just dichromats. They only have 2 types of cones. Some mammals, as well as some other vertebrates, have only a single type of cone, or perhaps no cones at all and only have rods. We'll take a look at a couple examples in a bit. But this system alone, this trichromatic system, does not explain all of the colors that we're able to see. So you might kind of think of a color wheel, how do we get colors like magenta and pink or brown even, and that are kind of combinations on the ends of the spectrum for, at least for the pink and magenta there. And what about colors like bluish yellow and reddish green? So why can't we see a bluish yellow or even conceive of it? What about a reddish green? And so this is where a kind of another theory of color vision comes into play called the color opponent theory. And so this is the idea that there's two opposing color pairs, red versus green and yellow versus blue. And all the other colors are kind of made up of combinations along these two axes. So we can't have greenish red and we can't have yellowish blue because they oppose each other. They're opposites. But we can have orange, which would be a yellow and red combination, or purple, which would be a red and blue combination. And so this theory also explains the effects of after images. So you might have stared at or done a test like this before. So just stare at that white dot in the middle, the white square in the middle. Don't worry about the moving ones. It's for a different test. We won't do that one, but just stare at that one in the middle there for another few seconds. And when I switch the slide here, you might see that after image effect. It goes away pretty quick, but you might have also noticed, and you might still see it, still see it moving around in your vision. So it actually tracks where your eye is moving, and you will see that the colors got switched. Right? So if you're staring at the same color for too long, the photoreceptors become fatigued, and then when you look away, the signal gets kind of mixed up in your brain. Right? And so that process is also explained by this color opponent theory. And this is still a simplification, and there's still some argument on the exact pathways here. We could kind of think of this as being split between a luminosity signal, so kind of our grayscale signal, and a chromaticity signal, so the color signal, and then having these kind of blue versus yellow channel, and then this green, versus red channel. Right, so this is how it works for us, for humans, for some old world primates that also have 3 types of cones. But for the typical mammal, use an example of a cat here, that would be a dichromat. So it just has a violet and a red photoreceptor. Something like a mouse, instead of the violet, that would be shifted over towards the UV. But other animals, including birds, a lot of lizards, turtles, actually have an additional photoreceptor type. Remember, the ancestral vertebrate would have had this number, 4 different types of cones. And so birds are tetrachromats, so they can actually see a wider color space than we can. Now the chicken here is not got a UV sensitivity. It's got a violet sensitive short wavelength cone, but a lot of birds do have UV sensitivity. And so we can kind of take a look at what UV vision might look like. So for human vision, we would just see this yellow flower. If we looked at it under only UV, it would look something like this. So brighter is more UV. If we simulated B vision, which has a UV channel, a green and a blue, it would look something like this. But then to the kind of bird, which has tetrachromatic vision, or at least this is a simulated version of it, it has a UV red, green, and blue channels. We could also look at some birds themselves, and they might use this UV to signal. Again, not all species of birds can see UV, but many of them can. And so here again, we have human vision, only UV, and then a simulation at least of what bird vision would look like. Now, as I mentioned, some vertebrates don't have any cones at all. These are often deep sea animals or fossorial species. So fossorial being species that spend the majority of their life cycle underground, so they're subterranean. And so this here is a caecilian. And so you might think it looks like a worm or maybe a snake, but it's actually a limbless amphibian. It only has one type of rod and does not have any evidence of cones, at least in the species we've looked at so far. So one of my students, Aaron, who's actually was here today, is studying these kind of fossorial adaptations in caecilians as well as in some squamate lizards. And so that's kind of a cool thing, but not really related to necessarily to color vision. So I'm not going to go into too much detail about it today, but these caecilians would have only the ability to see in grayscale and then only under kind of low light conditions. Presumably under bright light, their rods would get oversaturated. Now, if we look at teleost fish, they actually have way more types of photoreceptors than pretty much any other vertebrate. Now, does this make them, more than tetrachromats? Not likely. In most cases, they're not expressing all of these types. They're not using all of these types of photoreceptors at the same time. And instead, they actually might express them at different points in their life cycle, right? So as a juvenile, they might express some different, one or two of those green cones and one of those red versus orange cones. And then an adult, they'd use the other ones. And they even change them during change them seasonally. So as they kind of migrate, so some species like salmon are migrating from the ocean to the freshwater. So the available light is actually changing. And so they actually change up the visual pigments in their eye to kind of match that new environment that they're living in. All right, so I thought with kind of the rest of the time here, I'll go through a couple examples from other vertebrate groups, things that I've worked on. I don't know if I'll have time to go through all of these, but we'll take a quick look at snakes, geckos, frogs, and turtles. And so one of the things, as I mentioned, I studied during my PhD with snakes and geckos, and they're really interested in them because they had what we call simplex retinas. And so we saw most vertebrates have both rods and cones, and that made sense because most animals need to be able to see in the day and at night, right? So these simplex retinas have only rods or have only cones. And so I was kind of really interested. So this is in snakes. We have these nocturnal all rod, diurnal all cone, and we had the same situation, or very similar, I should say, independently evolved in geckos. I was kind of interested, why would you want to lose one or the other? Why give up that ability to be able to see in the day or be able to see at night, right? So how and why do these simplex retinas evolve? So if we look at snakes, we can break them into three main groups. We have scolifidians, blind snakes, and worm snakes. They have rudimentary photoreceptors. Hemiphidian snakes, pythons, and boas, they have these duplex retinas like we might expect, although they have lost additional photoreceptor types. But then in Cenophidian snakes, we actually have a great diversity. So we have duplex, all cone and some diurnal species. We have intermediate, where it's kind of in between, as well as some nocturnal species with only having rods. And so it was kind of postulated that there was this kind of transition from duplex all to diurnal all cone to intermediate to nocturnal. We've kind of shown since then in some of my work and others that this is not the case. It's not quite this simple. It's a lot more complex and a lot more transitions. But what I'll kind of talk about briefly now is, how did this all cone retina in these diurnal snakes evolve? And so there's two kind of hypotheses, kind of two ways this could happen. The first is that the rods were lost. And then we had a kind of split in one of the cone types, most likely the UV sensitive, into two different cone types. As we know that they have two small cone types, and then those two big cone types. And then the alternate way is that instead of the rods being lost, the rods are actually what we call transmuted or evolutionary modified into a cone. Right, so how could we test whether whether we have any support for this second hypothesis? Right, so we could look for the rod visual pigment, right, the visual pigment that's only found in rods. We could look for other proteins that are specific to rods, and we could look at the morphology of the cell and see if we can see any hints of morphology of an ancestral rod remaining. So with these images here, what I'm showing is immunohistochemical staining, which means we use an antibody to bind to particular proteins and then fluoresce that so that it would kind of glow out in a particular color. And so here, when they need the red color, is showing that the antibody bound to RH1, which is the rod opsin. So this is what we would expect if rods were not lost and were in fact still there. And then the green here is the rod-specific transduction. So this is another protein that's expected to be only found in rods. And again, found in these kind of cone cells, kind of blow it up there and see that they are overlapping in the same cells. And these are the cone-like cells that we expected. And so we found this in a different species of snake as well, and then a third species of snake, all of which are having, from a kind of gross morphological view, an all cone retina. All right, so we had support for our first two here. What about hints of rod morphology? If we look kind of broadly, so this is with a scanning electron microscope image, we can see that there's no evidence of any rods. But if we kind of zoom in, and so instead we're using transition electron microscopy, which is much higher resolution, you can see the kind of scale bar down there at the bottom, 100 nanometers, so quite a magnified image. That we do find small cones with the typical features we expect, which is these open discs. This is a feature found in cones where there's no membrane enclosing this side. But that some of these small cones, actually, once you blow them up a lot, they look a lot more rod-like, and they actually have the membrane along the edge here. So it's enclosing those discs. And so this is our kind of hint of rod morphology. We also did some other assays here with this kind of series of studies where we could demonstrate some cone-like properties of these cells as well. And so, but I'm not going to kind of show that now, but why cone-like rods? So why do we think this may have evolved? What kind of pressure might there have been that would favor changing the rod into a cone? So you can see with the Python example here, where it just has the two cones, there's a large portion of that light spectrum where there's very low sensitivity. Now, if we kind of fill that in with with the rod, if we make it function as a cone so that it can then be active, that could provide the basis for trichromatic color vision. So it definitely would improve spectral sensitivity in that region, but we think it might actually be based on pressure for increased color vision in these highly diurnal snakes. All right, so that was snakes. What about geckos? So geckos are mostly nocturnal, and they have these retinas that are all rods. And so ancestrally they were nocturnal, but a few different lineages kind of have switched to diurnality. And we think that for lizards, diurnality is actually ancestral. So we kind of think of this as a reversion back to the ancestral lizard condition of diurnality. Right? And these species tend to have retinas that look like they're all cones. We have all rod and all cone retinas in diurnal and nocturnal, or in nocturnal and diurnal geckos. And so again, we could put this in a kind of evolutionary framework of what we think happened here. And so we think that the diurnal squamate ancestor would have been diurnal, but with a duplex retina. And then at some point, the ancestor to geckos actually lost rods and it was left with only cones, but then became nocturnal along this lineage at some point and evolved all rod-like cells. Now, because it had lost the rods earlier, these would all be actually evolutionary derived from cones. And then we think that they reverted back to being an all cone, both kind of functionally as well as morphologically in a few of these diurnal lineages. And so there is some good support for this. And, some of my work contributed to this kind of transition here between the ancestral all cone and the nocturnal. But what I kind of wanted to highlight here, something that I think is super cool about geckos is that they have nocturnal color vision. So as we kind of talked about, most vertebrates just have a single rod and they're seeing grayscale under low light levels at night. But these geckos, at least in the species that have been tested, we think that they can actually distinguish color. So here's the kind of three light sensitivities, or the light spectra of the visual pigments, the absorption spectra of the visual pigments. I've kind of colored them here based on their visual pigments, which kind of doesn't match up exactly. So this one is the UV one here. The blue one is actually, I colored it green, and then the green one is actually the red. So it's a bit confusing there, but have the three types, which is the main thing. And so what Roth and Kelber did was do a behavioral test where they presented a gecko with two different crickets on forceps that were either colored with these kind of blue patterns or these gray patterns. So achromatically, so if you remove the color signal, these would look identical. So to us, under dim light, when this test was done, we couldn't tell the difference between the blue and the gray. We lose that ability, that chromatic signal under low light. Now they trained the geckos by providing a palatable cricket on the left and an unpalatable cricket on the right. So they actually just salted this one. The geckos don't like that. And so they don't want to take the gray one. They want to take the blue one. And so they tested them under low light levels, again, when we wouldn't be able to see the difference, and found that the geckos, much more frequently than would be expected by chance, chose the blue forceps with the palatable cricket. So this is good evidence that they can actually see, at least between blue and gray, see this color signal under dim light conditions when almost all other animals, besides frogs, which we'll talk about in a second, as well as some insects, then that can actually still see that color signal. How are we doing for time here? All right. Okay, so in frogs, as well as salamanders, they have a novel photoreceptor. So instead of having just a single rod, they actually have two types of rod, which we call the green rod, which is actually blue sensitive. So that's a bit confusing. And this one may also be involved in nocturnal color vision, at least in frogs. We don't have any evidence for this in the salamanders yet. But in frogs, there's been a behavioral study that looked at this where they compared green versus blue light and whether the frog could distinguish between those colors under dim light, right? Because these two rods are maximally blue and maximally green sensitive. And so what this graph is showing is that under complete darkness, there's no difference in the fraction of jumps toward green or blue, but that after we increase the light level even a little bit, we already start to see a difference between green and blue, and it actually gets stronger and stronger. And then for some reason, I don't remember why this was the case here in this study, but it switches. I think it's when the cones activate. And so then they actually prefer blue over green versus preferring green over blue. That must be the point where they don't mark that on the graph, but where the cones come into play. And then of course, those bit affect the color signal. So the cone phototaxis signal, meaning the movement towards light, is obviously different. But it's super cool that they could see down to all the way here. So this is actually like the visual threshold. This is about the point where below that, you really can't detect any light at all, let alone detect differences in the wavelength of light. And so I'm interested in how these green rods have evolved. So did they evolve from cones or did they evolve from rods? And so again, we looked at this with the snakes, but we could look for features of rod morphology. We could look for molecular rod visual pigments or other rod proteins that might suggest they have a rod ancestry versus having a cone ancestry. Now, some of this work has already been done. So we know that typical red rods, which we call them red rods, the typical ones have these closed discs. But we know, do we know if green rods have open discs? Right, this would suggest a potential cone ancestry. And then when we actually have looked at these, so this was done a long time ago with some electron microscopes, this is just an illustration of it, you can see that, they seem to be very similar in terms of their morphology. So they both have these closed discs. But where they do differ is in the inner segment. So the other part of the cell that's not actually involved in vision, but is involved in maintaining the cell itself, they actually have the red rods, the typical rods, differ from the green rod morphology, which is very similar to the cone morphology. So kind of a bit of a mismatch here. What about the molecular components? Do they have rod visual pigment? Do they have other rod proteins? Right, so this work was actually done in salamanders. So the red rods, the typical rods, they contain rod opsin and rod transducin, right? So that makes sense. What about the the blue cones first? They contain blue cone opsin and cone transducin. So both cone things. Again, that makes sense. But the green rods, that new type of rod, it has a cone opsin, but the rod transducin. So again, we have a bit of a disconnect there. Right, so we kind of don't have a great idea yet what the ancestry. We have this kind of mixed signal. And so one of the things we're starting to get going in my lab is using single cell RNA-seq to try to get at the bottom of this. And so we're going to try to target this chubby frog. It's a little hard to get a hold of, but it does have lots of green rods, which will make this a lot easier. Because when we dissociate the retina to try to get these cells so we can do molecular analyses with them, we won't be able to tell them apart anymore. So what we can do is individually sequence each cell using, there's lots of different methods to do that I won't get into. And then computationally, we can take apart the expression profiles of these cells and hopefully distinguish the green and the red rod populations, and then use that to identify the components that are unique to red and unique to green rods. And then, of course, we could combine this with some new microscopy studies. Right, so this will hopefully get us at the evolution of green rods, whether they're from cones or whether from rods. All right, the last thing I'll go over just very briefly is some work we've been doing in turtles. This is work done by a former PhD student of mine, as well as a bunch of other previous students, all the way back to some initial work I did during my PhD on this. So I'm sure you're familiar with turtles, but what you might not realize is that there's actually two main groups of turtles. We have Cryptodira, which are hidden neck turtles, and Pleurodira, which are side neck turtles. And they actually diverged about 200 million years ago. Now, the Pleurodira or side neck turtles are a lot less well known, especially where we are in North America, or where I am in North America. I guess some of you are in Australia. So they're actually found only in the southern hemisphere. And so kind of one of the most obvious distinctions between them is how they retract their necks, which is where the name comes from. So the Cryptodiras retract their neck into the shell, right? They pull it back there, like seen on the left, whereas the Pleurodires, the side neck turtles, fold their necks sideways. And the turtle visual system is actually really complex. It's as complex, or even in some ways, perhaps more complex, with more combinations of these oil droplet color filters that they have, which again, we're not going to have time to get into. They have these really complex visuals, so as are more complex than birds. But all the data that we have up to this kind of point is from the hidden neck turtles. And so we've done some genetic molecular work, comparing some whole genomes in these side neck turtles, as well as sequencing a eye transcriptome, so we can see what's actually expressed in the eye in some side neck turtles. Now, we've done a few different comparisons with this, some molecular evolutionary analyses, but the kind of result I'll highlight for you today is based on the SWS1, or short wavelength sensitive visual pigment. So hidden neck turtles, at least as far as we know, are all UV sensitive. But we found some mutations in the side neck turtle gene that suggest they're all violet sensitive, at least based on our kind of relatively small sample that we had. And so we kind of estimate the sensitivity of that being at 430, shifted from 371 in the UV. Right? So it's a fairly large shift. And this is important, right? So our kind of next step is to functionally assay the pigment, though, to actually determine if our estimate is at all accurate. So we can functionally assay this in the lab. And this has important implications for turtle behavior and conservation, because a lot of turtles are using that UV light to orient. So especially sea turtles are using it to orient towards the sea, but all the other hidden neck turtles are expected to be using that UV information, in their kind of life cycles. And so if there's a big difference between the side necks and the hidden necks where they're not using that UV signal, that can have pretty big implications on how they're interacting with each other and their environments, and how we might help to conserve them as well. All right, so there again is my lab, just to say thank you to everybody, and thank you for listening, and I'm happy to take any questions.
Great. Thank you so much, Ryan. Fascinating. Amazing that, you can have color vision at night and all these different variations of cone and rod. I have a leopard gecko. Do you know what kind of
Yeah, the
vision
leopard
it has?
gecko would have an all rod-like retina, and it likely also can see very well at night, and can likely see color at night as well. So very similar to the species that was shown is going to be very similar to your leopard gecko. Yeah.
Fascinating. Yes, we'll take questions. Jacob, you have your hand up. Go ahead.
Oh yeah, thanks so much. Oh, that was really fascinating. Thanks for sharing that with us, Ryan.
Thank you.
So I just have a question about the difference between rods and cones. So like what is the difference? Like definitionally, like what makes something a rod as opposed to a cone? You probably said this early on, but I forgot, because it's like I might have thought that the difference is that the cones are what allow you to see colors and the rods aren't, but then it turns out that you can use rods to see color. because there are some animals that do that. And so like what exactly is is it a functional difference? If so, what function is it? Or is it?
So there's a number of differences. The line is not, as clear as we make it out. We like to divide things into clean groups, but it's not really the case. But morphologically, they're different. So the rod is rod-like in shape. It's typically larger, although not always. It has these membranes that are enclosed by a disc, or so the discs inside the cell are enclosed by the membrane. But physiologically, they're much slower to react. They're much slower to recover, but they're much more light sensitive. So a cone photoreceptor will activate much more frequently just based on thermal radiation, right? So it doesn't necessarily need light to activate it. It will just kind of activate spontaneously. Whereas the rods have reduced that to the bare minimum of like the biophysical limit. So they barely activate in the absence of light. And so that's what gives them the really high sensitivity. There's also different pathways for recovering the cell. So I mentioned the retinal there. We need to get a new one in. And so there's a different cycle that supports rods and cones, and then a separate cycle that supports only cones. And that's partly what allows cones to move faster. And then those downstream proteins, I kind of didn't mention it too much, but there's rod versions and cone versions. And so there's all kinds of parts to this. And all these factors kind of contribute to the difference in the physiology that we see. And then developmentally, though, whether the rods are derived from a cone progenitor or something completely separate can also vary across taxa. But evolutionarily, we think it was a cone first, and then the rod would have developed from the cone progenitor.
So are there cases in evolution where it's really unclear whether you would want to call something a rod or a cone?
Yeah, so definitely those cases in the geckos and snakes, as well as this extra kind of rod type in the frog and the salmon, we still don't know for sure. So that's one of the things I would like to figure out in the amphibians. But in the snakes, we're relatively sure that the ones that have only cones, that's actually from a rod. And the ones that have only rods, most of those are going to actually be cones, although we haven't studied that in as much detail yet. So we still need to decide that for sure, as best we can at least. And then the geckos though and then there's a lot more research on the physiology there as well that helps kind of complement the fact that it's not quite as good as a regular rod or an ancestral rod. It's getting close, and we can look at some of the molecular changes that achieve that, and some of the other changes that achieve that downstream. But all of that kind of suggests it's not a real rod, if you want to say that, or not an ancestral rod.
Awesome. Thanks so much.
Yeah, for sure.
Any other questions? Lauren,
Lawrence.
go ahead.
Thanks. Yeah. No, that was a great talk. I really want to come and visit your lab at some point.
Yeah, for sure.
I just wanted to ask a quick question about the turtles. For the cones in the turtles, are they all the same except for the oil droplet? They change only because of the properties of the I suppose the chemicals in the oil droplet and the cells themselves being the same. Is that true?
Only for a couple of them. So most of them are going to be the same differences that we saw, between all the other pigments with those curves. So, but there's some additional types there that have the same visual pigment, and the only thing distinguishing them is the oil droplet. And so that gives them the potential to be beyond tetrachromatic. Whether they actually can process that downstream is kind of, up for debate still, or up for, us to figure out. But those oil droplets act as color filters, long wavelength color filters. And so, they can control what light is actually reaching the visual pigments. And so then, of course, can potentially increase color discrimination. There's other ones that are colorless too, and they may just be focusing light to help kind of capture as much photon capture as possible, focusing them on the visual pigments. But the turtles are, I think, the only example where they have a cell type with the same visual pigment, but two different oil droplets that actually differ in their absorption properties and transmission properties.
Are there other species that use oil droplets then? You sort of implied that, or not?
Yeah, so birds will also have them.
They have, okay.
Lizards have them. Some fish have things like them. Some other taxa have micro droplets, so they're not quite the same, but they're maybe serving a similar function in some ways, but not the color pass part. But yeah, mostly it's just mammals lost so much of these things. So oil droplets may also be even ancestral, although I wouldn't go so far to say we have great evidence for that yet.
Very interesting. Thank you.
Thank you.
Any other questions? Ryan, in terms of, a species being all rods or all cones or variations, are there certain evolutionary pressures that might have pushed one to develop one way versus the other?
Yeah, so the one example I talked about was in the snake. So we think maybe it was fill in that gap pressure, right? So we're missing a lot of that spectrum. In geckos, it's harder to think of what's the pressure that drove them to lose their rods initially, because that's a little bit odd. So that one I'm still puzzling on, and I've been puzzling on it since I started my PhD, I guess, and scared to get into this. Why did they lose rods initially? And then after losing rods, what pressure then drove them to become nocturnal? Seems like if you've already committed this much, why would you then go back? So that one is still a huge mystery to me. The transition from the nocturnal to diurnal makes a lot more sense because if you're shifting to being active primarily in the day, then shifting all your cells to function better in bright light, right? Meaning reducing their sensitivity, increasing their reaction speed and recovery speed. That makes a lot of sense. So that part is great, and that's the part is easier to study than what happened in the past that we can no longer actually get to. But in some ways, that's the part that's more interesting. So like, why? So that one I don't have a great answer to. In the frogs, what would be the pressure to develop a second type of rod to allow them to see color? Likely mating, right? Likely to try to see some of those small color variations that they might be using as mate choice signals, being able to see those under dim light when most frog species are active and actively calling and looking for mates. And so again, that might be what would drive that. In salamanders, we don't have good evidence that they necessarily could be used for color vision at night. And so whether that's the same evolutionarily the same cell type, we would assume just based on parsimony that it's the same, but we don't know that for sure yet. We don't have any good evidence to support one way or the other than parsimony that they must be the same. But whether they serve the same function, we have no idea.
Such amazing diversity.
Oh yes.
I just have another quick question. They're finding that more and more animals are fluorescing.
Mhm.
Would that somehow maybe impact some of this visual changes, or how would the fact that they fluoresce impact their visual system generally?
So yeah, biofluorescence is like a kind of newer research topic in this kind of field. Whereas one wavelength is absorbed and another wavelength is reflected or transmitted out, or is fluoresced out, I should say. So that would just shift the color pattern on the frog or on the whatever what we're looking at. I'm most familiar with this in frogs because there is a researcher that kind of we've been chatting with that's studying biofluorescence and is interested in this. And we've been kind of sharing some of our data on the absorption spectra of the visual pigments so that she can match it up with what they're fluorescing and see if there's any kind of correlation there. So jury's still out on that one. Hopefully we'll have some idea soon whether there might be some match there. Kind of my thought on it is, how strong can that fluorescent signal be? Can it make a real difference under kind of dim light conditions when frogs would be active? I don't know about that. A lot of the images you see about fluorescence, people are shining really strong narrow band wavelengths onto the animal and getting it to then fluoresce. But that's obviously not what's going to happen in nature. So whether it's just a byproduct of the pigmentation that they're using in their skin that happens to biofluoresce, or whether there's actually selection pressure for that fluorescence, I think is a cool topic, but it's one that we don't know enough about yet.
Great. Yeah, because chameleons and fish, and there's all kinds of it that they're finding.
People are doing more and more tests, and every time they test a new animal, they're finding more versions of biofluorescence, which is super cool.
Yeah. Paria, go ahead. You had your hand up.
Thank you. That was a very fascinating talk. I'm wondering if you have studied processing of a single color beyond the retina, and if like processing of a color in the brain of the animals would answer some of the questions that we have about the rods and cones and the distributions and all that.
I have not done that. I definitely think it would though, and it's always kind of like in the long-term goal, right? So, we're focused on even just the visual pigments, and we're trying to expand now to the phototransduction process. And obviously, eventually, we'd like to expand through the rest of the retina and then through to all the visual centers in the brain. But we're just not there yet. But it is a great goal, and yeah, we can't just look at one or just look at the other. You're not going to get the whole picture. So that's a good thing to always keep in mind.
Thank you.
Thank you. Yeah, opponency. How would you do opponency and with that many cones?
Yeah, you can measure it at the retinal level, and then you can measure it at the brain level, and yeah, some collaborations in the future to look at this would be great.
Do you know Kasim Zaidi from SUNY in New York?
I don't know.
So he had a paper about particular kind of shrimp with 12 types of cones. And
Oh,
the
the mantis
idea
shrimp,
that
yes.
and then like what they were talking about in their paper was that because they don't do any kind of high level kind of processing in the brain, so the shrimp will require 12 kinds of cone to have a good resolution of all the different wavelengths. Whereas in humans, because we have this hierarchy of visual areas, then we will be able to generate span all the wavelengths and then generate a selectivity to particular kinds of hues and so on. And that's why only 3 cones are enough in humans. Yeah.
Exactly, yeah.
Yeah.
Great. Well, thank you so much, Ryan. A round of applause.
Thank you. Thanks for
Very
having me.
fascinating talk, and yeah, just mind-blowing, all the diversity and the way, rods, seeing in color with rods is amazing.
Yeah, super cool.
Thank everybody for attending. I just want to point out, stay tuned to our website. I put it in the chat earlier. We have another talk February 11th. Laura Berman's a printmaker, going to talk about color and printmaking. And we have our student awards that some of you may be interested in. The deadline is March 1st. And again, check out our website for that. So thank you again, Ryan. Very fascinating talk. Really, really interesting work. Congratulations.
Thank you.
And
thanks for inviting me to give this talk. It's been fun.
Great. Thank you so much. And thank you everybody for joining us. And please check out our website. Consider joining us as a member to the CRSC so we can promote a very diverse view of color and interactions and multidisciplinary lens on color. All right. Good night. And we'll see you at the next one.
All right. Take care, everyone.
Bye-bye for now. Take care.